Hey class, hey Dr. Crum. Hope everyone is happy and healthy and doing well. These online classes are definitely weird and gonna take some getting used to. My question was Watson and Crick proposed models for DNA replication. They proposed 3 models. One was correct, and it was proven by Matthew Meselson, Franklin Stahl, and Jerome Vinegar. They proved, or they were able to find out, that semi-conservative DNA replication was the correct method. Now, the first one that Watson and Crick proposed was the dispersive method, and that is where they take chunks of the parent cell. Ooh, let's figure out how we can do this. All right, we can see my Picasso drawing. As we come here, this is a dispersive backwards. And what we do is if this is written in pen, and this is breaking down, this is breaking down into pen and pencil, and it's broken down into segments of pen, pencil, of double helixes that shows the separation or the new replicated helixes that are coming in bits by bits of however many nucleotides at a time. And that is how the dispersive replication method happened. My great backwards drawing. Now, the other one that they proposed that was incorrect was the conservative method. And as the lighting can tell, I'm limited on school supplies, of online school supplies. The conservative DNA replication method had the parent DNA double helix strand, and it is just completely replicated. So here's the parent, and then here's the new one in the pencil, and it is just a brand new strand that has been replicated. Now that was proven incorrect, as the semi-conservative is the more correct method, as you have one strand that is from the parent double helix, and you have the other one that has been replicated and added on to form the new double helix. And that is what we have now learned as our new DNA replication. Now with my awesome drawings to help you guys out, that kind of breaks down how you can show DNA replication on a very, very, very basic level. Now, how they found out that it was semi-conservative DNA replication is very, very interesting. They had 2 different types of nitrogen isotopes, and it was nitrogen 15 and nitrogen 14. And they grew a culture of bacteria in a nitrogen 15 environment. And this culture's DNA was taken, and it was started to now grow in a culture of nitrogen 14. And this culture was then grown, and they took the DNA of the new generation of these combined cultures, and they put it into an equilibrium density centrifuge. And an equilibrium density centrifuge spins these vials until it separates it very finely, or very accurately, very precisely, of separate DNA, even based off of density, equilibrium, density, centrifuge. So it's broken up, and you can then find out the ratios of how much nitrogen 15 and nitrogen 14 has been layered in this vial that has been spun for time on time again, and it is for such accurate and precision that it must take hours on hours on end. And the ratios that were found after combining the N14 and the N15 cultures showed that it was semi-conservative, and it was able to show the weight distribution or the density distribution, that there was N14 and N15 combined in the separate new and parent cell or strands combined. So if this is it coming off and then it spiraled off here, this was proven based off of the ratios. That's kind of their experimental method. It's pretty interesting how they did it. That's pretty much the rest of my question, and I really do appreciate it. But for listening, everybody, and hopefully I didn't make a complete fool of myself, and I hope you guys are finding things to do and holding your family tight and loving every minute of this break and online stuff. So take well.